Hello everyone. Today I will be presenting a topic on apoptosis. So let's begin. What is apoptosis? Apoptosis is a programmed enzyme-regulated cell death that occurs mainly in multicellular and in some eukaryotic single-celled microorganisms. Um, During apoptosis, a cell undergoes several characteristic changes, such as nucleus and cytoplasm degradation. Um, after the nucleus and cytoplasm is degraded, the cell shrinks, and its contents are placed into small membrane-bound vesicles called apoptotic bodies. Uh, Once the apoptotic bodies are formed, in order to prevent the release of potentially harmful intracellular components, um, these vesicles, apoptotic bodies, they are cleared away by phagocytes, um, which digest them. So yeah, next, why do we need apoptosis? Um, this process is actually essential for many reasons, and one of the main of them is organ development. So during embryonic growth, apoptosis helps shape our fingers and toes by removing the webbing between them. So by removing the unnecessary cells between them. Yeah, and it helps to eliminate damaged and mutated infected cells that prevent the development of cancer and tumors. Um, in our immune system, once immune cells have fought off an infection, apoptosis removes those cells in order to prevent the excessive immune reactions. And also, above to all of this, um, in addition to all of this, apoptosis maintains homeostasis, so it maintains a balance of cells in our tissues. Yeah, so you can see from the pictures here, yeah, organ development, cancer cells, and also immune defense. Yeah. Okay, so next, what happens when apoptosis is unregulated? So when apoptosis is unregulated, it can cause some serious disease. So too little apoptosis allows abnormal cells to survive, and this can cause cancer and autoimmune disease. Um, but too much apoptosis also can cause some disease. It can destroy too many cells and lead to neurodegenerative disease like Alzheimer's and Parkinson's, and or also to immunodeficiency disorders like sepsis. So what are causes of apoptosis? Apoptosis can be triggered by internal damage such as DNA mutations or oxidative stress, external factors like cytokines and hormones. Also, it can be triggered by immune responses, where cytotoxic T cells target infected cells. Uh, in development processes, apoptosis also necessary, and in loss of growth factors also can cause apoptosis. Next is morphological changes. So before the apoptosis occurs, the cell undergoes several morphological changes. First of all, cell shrinks and its cytoplasm becomes dense. Then chromatin starts becoming dense, and this process is called pycnosis. After the pycnosis process occurs, DNA starts breaking down, so DNA fragmentation occurs, and this process is called karyohexis. After this, cell surface starts blebbing, and it forms vesicles, which later turn into apoptotic bodies. Yeah. So yeah, our next thing is caspases. Caspases are crucial enzymes that carry out apoptosis. They are actually similar in its function to proteases and enzymes that break down proteins, because caspases also break down, cut up important proteins, and that lead the cell to die. So we can divide caspases into two main groups: initiator caspases and executioner caspases. Initiator caspases, such as caspase 8 or caspase 9, they actually begin the apoptosis process. They activate the executioner caspases. Executioner caspases, examples of them are caspase 3 and caspase 7. They destroy key cellular components and actually execute the cell death. Next is apoptosis occurs in three distinct pathways. May occur in three different pathways. First of all, the intrinsic pathway. It's mediated in mitochondria. Then extrinsic pathway. It's DAS receptor mediated pathway, and also perforin granzyme pathway. It is initiated by immune cells, and it's also may be considered as extrinsic pathway. And let's look at each of them in detail. So, intrinsic pathway. In the intrinsic pathway, mitochondria becomes leaky and ooze out proteins called cytochrome C, which actually initiate apoptosis. So usually the cytoplasm and mitochondrial membrane, they contain these proteins called BCL2 and BCLX, which are anti-apoptotic proteins. They preserve the integrity of the mitochondrial membrane and prevent apoptotic proteins like cytochrome C to leak into the cytoplasm. However, when the death signals come to the cell, for example, the DNA is damaged, or proteins are misfolded, or there is an oxidative stress, these BCL2 and BCLX proteins become blocked by apoptotic proteins called BH3 only proteins. They block the BCL2 and BCLX proteins, so the cytochrome C leaks into the cytoplasm, and in the cytoplasm it binds with an APAF1 proteins, apoptosis activating factor 1 proteins, and together they form an apoptosome. This apoptosome complex then binds with caspase 9 and begins to cleave to activate other caspase 9 enzymes. And together, all of them, they activate executioner caspases, caspase 3 or caspase 6, and this leads the cell to death. Yeah, so from the picture, from the diagram here, you can observe it on your own, how this process occurs in details. So next is the extrinsic pathway, or DAS receptor mediated pathway. This pathway is initiated by external DAS signals, for example, infected or cancerous cells. Specific ligands, such as Fas ligand, bind to the DAS receptor located on the target cell's surface, like the Fas receptor. Once the ligand binds, it triggers the formation of DAS-inducing signaling complex. Shortly, it's called DISC. This complex includes adapter proteins like FADD and also procaspase 8. In the DISC, procaspase 8 is activated to caspase 8, and then it activates other executioner caspases, which leads the cell to die. So from the picture here, you can observe this process as well. So our next is granzyme perforin pathway, or immune-mediated pathway. This pathway is used by immune cells, immune system. So the immune cells, like cytotoxic T cells or natural killer cells, when they detect an infected or cancerous cell, they release perforin, a special enzyme that forms a holes port in the target cell's membrane. Um, then, after the pores are formed, they release granzyme proteins, which enters through the spores, and they initiate apoptosis in those cells. So usually there are two different types of granzyme proteins: granzyme A and granzyme B. Granzyme B activates caspase 3 and leads to apoptosis through the classical caspase way. But granzyme A, it induces a caspase-independent death pathway, but it also leads to the DNA fragmentation and causes apoptosis. Yeah, so additionally, you can observe here some diagrams where this process is illustrated in details. So to conclude our presentation, we can make several notes. So apoptosis is a controlled non-inflammatory form of cell death. It relies on caspase enzyme to execute the process, and there are three main pathways of apoptosis: intrinsic, extrinsic, and perforin granzyme pathway. And finally, apoptosis is really necessary for organ development, cell balance, and disease prevention. Thank you for your time. Thank you for listening. I hope this presentation was useful and helped you better understand the importance and mechanisms of apoptosis. And if you have any questions, please feel free to ask. I'll be happy to answer them.